Good day, ladies and gentlemen. Today we're going to tackle ethical issues of embryonic stem cell therapy. So, as the world advances and as the medical field progresses, many kinds of researches, experiments, interventions, and anything you name it are rising everywhere around the planet. And one of these is this ESC therapy. But what exactly is ESC? How does it work? How much does it cost? And is it an ethical procedure in the Philippines and to the rest of the world? Every questions asked will be answered right here, right now with the Group 5 in Blessed section.
Embryonic stem cells are the key to embryonic stem cell therapy. These cells can be described as the foundation of all other cells. Embryonic stem cells are obtained from a human blastocyte, which forms 3 to 5 days after an egg cell is fertilized by a sperm. These cells are known as pluripotent, which means that they allow for the development of more specialized cells, which are crucial to the development of all organs and extremities in the human body. Embryonic stem cells are the building blocks from which more specialized cells originate from. This is also the only cell in your body that has the ability to generate new cell types, and under the right conditions, these cells divide into new stem cells called daughter cells. From then on, they can specialize into different types of cells such as nerve cells, cardiac cells, liver cells, blood cells, etc. This process occurs during the development of a fetus and is a process called differentiation. Under lab conditions, the process of stem cell daughter creation and also the stimulation of these daughter cells into specialized cells can be replicated. This concept of lab-created specialized stem cells and the ability to create more daughter cells is the cornerstone of embryonic stem cell research. So embryonic stem cell research is possible because of in vitro fertilization, which is a process used to help couples with fertility issues attain a pregnancy. In simple terms, this process takes place through the retrieval of fertilized eggs from the mother, which are then fertilized by sperm in a lab setting. And by the end of this process, there are many 1-week-old embryos left, which are no longer needed and will most likely be discarded. And these are the embryos which are donated to scientific organizations for the purpose of stem cell research or embryonic stem cell therapy. So the concept of embryonic stem cell therapy is a product of research which began in the year 1902. The Russian histologist Alexander M. Maximov was a driving factor in the history of stem cell therapy. He identified stem cells as polyblasts and also proved that all blood cells mature from a precursor cell with his confirmation of the Unitarian theory of hematopoiesis. From his research, many scientists from around the world began studying stem cells through animal, plant, and human experimentation. So what is stem cell therapy, now that we know all the background information about it? Well, stem cell therapy is a form of regenerative medicine that has been derived from this extensive field of research. This is an idea with extreme potential for the combating of many different diseases. This is because embryonic stem cells keep their pluripotent state if the correct conditions are maintained. In a lab setting, this can be achieved for long, if not indefinite amounts of time, as scientists are potentially able to grow millions or billions of embryonic stem cells because these cells divide about once a day. These lab-produced stem cells would then be able to be stimulated into differentiation, which would allow for the development of many different cell types and consequentially allow for the treatment of a broad range of degenerative, malignant, or genetic diseases, as well as treat injury due to inflammation, infection, or trauma. Because this type of therapy has the potential to treat such a wide variety of disease, its therapeutic potential is highly valuable.
Okay, so how embryonic stem cells perform? Embryonic stem cells are derived from inner cell mass of the blastocyst and place this cell in culture dish with nutrient-rich fluid that give rise to embryonic stem cells. Scientists can induce embryonic stem cells to replicate themselves into an undifferentiated state for a very long period of time before stimulating them to create specialized cells. So how the cell lines can be cultured and how to stimulate the stem cells to differentiate. Cell culture is a term that refers to the growth and maintenance of the cells in a controlled environment outside of an organism. A successful cell culture is one that keeps the cell healthy, dividing, and specialized. The culturing of stem cells is the first step in establishing stem cell line, a propagating collection of genetically identical cells. And once they have established a stable stem cell line, scientists start the process of causing the stem cell to differentiate into specialized cell types. The cellular environment in which stem cells naturally resides provides scientists with clues about how to make them and differentiate into a culture dish. In a culture dish, it will be essential that scientists are sure that stem cells have fully differentiated before they can use them for medical applications. So what are the sources of embryonic stem cells? First is in vitro fertilization or IVF. So what is IVF or in vitro fertilization? This is a process of retrieval of woman's egg via surgical procedure after undergoing an intensive regimen of fertility drugs. This is to stimulate ovaries to produce multiple mature eggs. And when IVF is used for reproductive purposes, doctors typically fertilize all the donated eggs in order to maximize their chance of producing a viable blastocyst that can be implanted in the womb. So, for the next sources of embryonic stem cells, this is the nuclear transfer. So what is the nuclear transfer? This is a process of inserting nucleus of an already differentiated adult cells, and for example of this is skin cells, into a donated eggs that has had its nucleus removed. The stem cells that are created in this way, therefore copies or clones. And lastly, nuclear transfer has not been successful in the production of human embryonic stem cells. So what is the differences and similarities of in vitro fertilization and nuclear transfer based on their ethical concerns? So in vitro fertilization and nuclear transfer are both has a process of destruction of human blastocysts, and this blastocyst can no longer has the potential to become fully developed human being. In vitro fertilization and also the nuclear transfer, they also require an informed consent to the donators of the blastocyst and also to the eggs, and this donator should be volunteer. For the last one, nuclear transfer is concerned about misapplication for reproductive cloning. As I have said, nuclear transfer is sometimes misinterpreted as reproductive cloning. So what is reproductive cloning? Reproductive cloning is widely used technique for implanting human animal embryo of an animal. So this reproductive cloning should not be practiced into human embryo because it is dangerous and it is most likely to fail.
Nope, no. According to Felongco 2012 and the DOH itself, the Philippine government has taken a firm stance against the use of aborted fetuses or human embryos for stem cell treatment, while also cautioning against the usage of such new medical treatment and or therapy. Though we may hear and see a lot of researches for embryonic stem cells available in the Philippines, still the DOH said that the procedure is still in its development stages and there could be ethical questions surrounding its use in the country. But traveling across the seas and lands, we have different countries who are already applying these interventions in animals, which is a little bit closer and leading to human for experiments. These are Japan, Singapore, which is also known as the Asia's stem cell capital, China, South Korea, Australia, South Africa, United Kingdom, Switzerland, Brazil, United States, and Mexico. Some of them prohibits reproductive cloning, but those countries are already accepting researches and experiments and treatments in terms of ESC therapy. But how does it cost? How much does it cost? Stem cell therapy cost can range anywhere between $5,000 to $50,000, or in the Philippine peso, it is equivalent to 2.3 to 2.5 million pesos.
Good morning, everyone. I am Francesca, and today I will be discussing the advantages and disadvantages of embryonic stem cell therapy. Through cell division, embryonic stem cells have the potential to divide and differentiate into a variety of specialized cell types, allowing them to regenerate. They are crucial to the development, growth, maintenance, and repair of our brains, bones, muscles, nerves, blood, skin, and other organs, according to GQE. Embryonic stem cells have a lot of significant advantages, including the ability to treat hundreds of diseases and a wide range of medical possibilities. The creation of embryonic stem cells, particularly for research purposes, can be done with the in vitro fertilization method. This enables researchers and scientists to focus on specific genetic features relevant to the study of specific diseases. For example, research employing embryonic stem cells could lead to studies of the origins of inherited diseases like cystic fibrosis, using stem cells from egg and sperm donors who have this disease. Embryonic stem cell therapy prevents the embryo from being destroyed for no reason. They are made from the excess embryos from in vitro fertilization. These are being used and experimented in stem cell research laboratories. This ensures that these embryos are not destroyed without a cause. Instead, they serve a useful purpose in medicine. Cancer research will make significant progress with the support of embryonic stem cells. They have a lot in common with cancerous cells, and this could have a huge impact on the lives of a lot of people all over the world. In addition to the advantages of embryonic stem cells, these are more flexible than stem cells found in adults. Adult stem cells are considerably more difficult to harvest, purify, and retain in the laboratory, according to Heinz 25. Embryonic stem cells may be used to treat diabetes and heart disease, which are both quite common and affect a large number of people in society. The primary downside with embryonic stem cells is the technique that they are obtained. When the blastocyst is used for the stem cell research, the inner cell mass is removed and the cells are placed in a culture dish. The blastocyte no longer has the potential to develop into a human being once the inner cell mass is removed by the scientists. In other words, human embryos are being destroyed as embryonic stem cells are being harvested for a study. Life, according to prominent philosophers and other people, begins at conception, with the egg is fertilized with the sperm. Embryonic stem cells involve the intentional creation of blastocysts that will never develop into a human being, which is seen as a controversial ethical issue. One of the drawbacks of embryonic stem cells is the high expense of the procedure. Harvesting a single embryo might cost up to $2,000 per occurrence. Another disadvantage of the therapy is that it has a high rate of rejection. It has been known that this therapy can lead to a variety of long-term health difficulties, including a negative immune response, tumor development, and growth of unwanted tissues, as explained by Yale. Another problem is that no one knows everything there is to know about embryonic stem cell therapy right now. Experimenting with blastocyst embryonic cells is deemed immoral by people who think that life begins at conception and that an embryo is therefore a person.
According to Kant's duty ethics, when it is your duty, it is morally right even if the consequences of that action is right or wrong. So when it is a duty to research about the embryonic stem cell therapy, or to do the embryonic stem cell therapy, you are doing a moral action, even if the consequences are bad or good. Based on Kant's duty ethics, if it is for one's own personal reasons or inclinations, then it becomes an immoral action. So if a person is doing it based on their duty, their action is morally right. Next is for natural theory. So in natural theory, the procreation should not be hindered. So the embryonic stem cells that are being harvested are potential life, or can be called life itself. So if the embryonic stem cell therapy or stem cell research prevents a form of life or a potential form of life to have no chance of living at all, it is an immoral act. The embryonic stem cell therapy will become an immoral action. Next is for the utilitarian theory. For the utilitarian theory, the goodness for the majority of the people would be the chosen action. So if there would be 100% cure for all the diseases that the stem cell therapy claims it can cure, then for many it would be very helpful, especially the crazy diseases, like the crazy claims of diseases that says that it can cure. But also we must put into mind that they call the embryonic stem cell a form of life. Some call it a form of life already. So is the majority going to benefit in the sacrifice of the stem cells, or the stem cells are the majority because those who will benefit are only few? And lastly is the justice theory. So in justice theory, everybody is balanced. Everything is balanced according to the law. Everyone has the right, and if the stem cell is already alive, they have a right for themselves. They must be judged, and in justice there always must be a limitation. What the limitation for this research is, and also what's the scope of the research that cannot be do, and the things that can only be done. So in justice theory, the law has a blindfold and everybody will be judged equally. Thank you po.
Now, for the advantages and disadvantages, or what you call pros and cons, our group gathered several articles to discuss both sides about the term. First, for the pros, written by Habib and Gordon in May 2006, with the title of Clinical Application of Stem Cell Therapy. He stated that these stem cells have the potential cure in a wide range of degenerative illnesses. So those diseases or illnesses that affect tissues and worsen in a matter of time, ESC or embryonic stem cell can be its possible cure. Osteoarthritis, osteoporosis, and Alzheimer's diseases are the examples of this. Next is the article written by Douglas and Savulescu on 2009, which was entitled Destroying Unwanted Embryos in Research. The study shows that it elevates suffering, lengthens lives, and allows parents to achieve their reproductive goals. This may also help to close the growing gap between organ demand and supply and to improve transplantation success rate. Lastly, for the pros, we have the article entitled Stable Benefit of Embryonic Stem Cell Therapy in Myocardial Infarction, written by Hodgson, Befar, Zingman, Kane, Perez Terzik, Alexev, Pouquet, and Terzik on 2013. The current investigation of myocardial infarction reveals that embryonic stem cell therapy has a consistent positive impact. This showed up as a long-term improvement in cardiac contractility and verticular remodeling, as well as verified cardiogenesis in the infarct zone from the implanted stem cells. This data suggests that the benefit of embryonic stem cells delivery begins early, as seen in the current study at 3 weeks following therapy, and is unaffected by spontaneous failure of stem cell-derived cardiomyocytes or host rejection of this allogenic transplant. The lack of declining effect over time suggests that embryonic stem cells could be used therapeutically in a long-term management of cardiac damage. Moving on, for the cons or disadvantages, we also gathered 3 articles from different resources and authors to see the other perspective of ESC or embryonic stem cell. First one, the article entitled Against the ATS Statement on Human Embryonic Stem Cell Research, and it says that despite of the characteristic which they can see in embryonic stem cells that they can be utilized to treat a variety of individuals, still there are major ethical concerns and the very likely possibility that because they are immoral, they will become tumors after being transplanted into patients. But these hurdles to widespread adoption will, without a doubt, be overcome in the future. Next is the embryonic stem cell research, an ethical dilemma by Wigwe, 2019. It says that because an embryo is considered a human being with moral rights, the principle of beneficence is broken, and the embryo's autonomy and welfare are not respected as a result of embryo harvesting in stem cell research. It also breaches the embryo's autonomy and human rights, as well as the autonomy of embryo donors and pro-lifers. Embryos are not treated as ends, but when they are killed to collect stem cells for research, they are treated as ends. Hence, this violates the principle of beneficence and utilitarianism. And lastly, the end never justify the means. We will all die at some point, but that does not give a researcher the authority to end someone's life. The embryos will die in this situation because others have chosen to kill them for the purpose of research. That is according to Thomas Jr. and Vasallo, which wrote against the ATS statement of human embryonic stem cell research on 2019.
I'm here to report about our individual stands, a summary of our individual stands regarding our topic, embryonic stem cell therapy. So we conducted a vote in our group, and our group had a vote of 4-1 for against embryonic stem cell therapy and 1 supporting embryonic stem cell therapy. So the main points of the 4 who are against the embryonic stem cell therapy is, first is, it is like playing God because of all the crazy diseases that it claims that it can cure. Next is destruction of human life or the embryo. So a destruction of human life or a potential human life. Next is embryonic stem cell therapy is so expensive that only the rich can afford when it is released to the public or accessible to the public. Next is the embryonic stem cell therapy have not published one successful outcome of its research. And for the support, the main points of support is potential cure for different degenerative illnesses, advancement of medical research, and it also opens different opportunities and discoveries for medical field. Nothing was violated since every procedure requires informed consent from all the cell owners. And that is our vote 4-1. Thank you.